All right, guys, we are going to talk about enzymes today. So here are the major facts, all right, and highlight along with me. Okay, enzymes are protein molecules, and because they are protein, they are going to be built of and made of amino acids. Okay, enzymes are going to bind to other molecules to help to digest and or synthesize molecules in the body. We call enzymes biological catalysts because they speed up chemical reactions. And I like to think of this as catapult. Be clear, they do not give energy to reactions. They just lower the amount of energy that's needed in order to do it. What's really important and might be misunderstood is that an enzyme is required for every single chemical reaction that happens in the body at every single moment. From doing cell respiration, to thinking, to moving, to chewing, to breathing, to everything that we do, we need a chemical reaction for. Enzymes are very, very, very specific in shape, like a lock and key, in order to fit whatever molecule that they're working with. And lastly, enzymes are produced in the ribosome, which is an organelle inside of our cells that makes proteins. So let's take a moment just to make sure we understand what digestion looks like and how we can identify digestion versus synthesis. So obviously we've talked a lot about this, but digestion is breaking molecules in the body. So enzymes can break apart bonds in large molecules. This is called digestion. And I want you to take a look at this picture right here. We see that we start with one item, this complex sugar, and then it breaks down into two. So that is how you know it's digestion. I want you to take note as well that this catalyst right here, and you might want to actually label that. Okay, the enzyme is right here. How do we know it's labeled catalyst? Okay, so sometimes they'll use the word enzyme and other times they'll use the word catalyst. So synthesis is the opposite, right? That's when we actually form bonds or we kind of glue molecules together to build larger things. So how do we know this is synthesis? Well, we start with two things. We end up with one thing because they were put together. And if you want to label the catalyst as enzyme, go right ahead. But that is also the enzyme. I have a couple pictures there too for you to kind of look at and read that could help you understand digestion and synthesis. One big example of digestion, and is not the only way that enzymes digest, is breaking food down in your stomach, where your stomach secretes enzymes or your mouth secretes enzymes. And some examples of synthesis that I'm kind of reading from here is building muscle, fat, and bone and hair, basically building your body. So let's take a look at the components of a chemical reaction. Okay, so how do we identify the enzyme? The enzyme pretty much is going to remain the same. It's going to be the only thing in the reaction that remains the exact same. So if you look over here, this kind of blue enzyme, it didn't really change, didn't break apart, right? So that's how we know that's that catalyst that's going to actually make or break the molecules, right? The substrate is whatever molecule that the enzyme is working on. So this could be a sugar, it could be a protein, it could be a lipid. So that's this substrate right here. And that substrate will get changed in the reaction. It will get changed into a product or products. And last but not least, the active site is where that substrate will actually connect to the enzyme. And that is that perfectly fitting shape, right? Like kind of that lock and key when the key slides right in with no resistance. So that active site is actually where they bind together. And that active site is really important because the shape has to be very specific. All right, let's dive into the structure and function of enzymes. So we're really talking about that specificity of shape, okay? Not only are enzymes very specific in shape, they also are all different from each other, depending on what they work on. So if we look right here, okay, this enzyme has this substrate and has a way, way, way different shape than this enzyme, right? This enzyme can't do anything with this substrate, right? It just literally wouldn't fit it. So these enzymes are going to be for two different reactions. Let's look at an example. Okay, so we're kind of reading down here. So for example, the enzyme amylase in the saliva of your mouth, it has a specific shape that fits amylose sugar. However, because it has this specific shape that fits amylose, it cannot break down any other sugar. It can't break down the lactose or sucrose or lactose. It can only break down amylose. So we have specific enzymes for specific molecules. We also like to say that the job is determined by the shape of the enzyme. Let's look at one more example, a couple more examples, right? Another example might be when you build keratin in your hair. You make an enzyme that has a specific shape for building keratin protein in your hair. And so that enzyme can't, work in your stomach and break down food because it's only built to fit keratin protein and synthesizing those molecules to make hair. This is like a lock and key because although my key and your key have the same job, opening something up, they have very different shapes and are only able to fit a specific door. So if we look at these two enzymes here, we have lactase and maltase. They're both breaking down sugars or synthesizing sugars. However, they have a completely different shape, right? So here's lactase, here's maltase, and they have completely different shapes that only fit the sugar that they work on. All right, so we're going to look at some specific examples of enzymes during digestion and synthesis of our macromolecules. As a reminder, carbohydrates are built of simple sugars, lipids are built of fatty acids, and proteins are built of amino acids. So when enzymes digest and synthesize carbohydrates, here's some pictures I've seen come up on the Regents exam demonstrating this. So notice that digestion is breaking something down. Okay, we're going to label X as the enzyme. We're going to label starch as a carbohydrate. Right? And if this enzyme is breaking down this carbohydrate or this big starch, we're going to produce simple sugars because those are what make up starch. Now let's look at synthesis, right? So in the opposite way, we have these four blocks coming together in this X thing, right, above the arrow to build starch. So the first thing we should realize is this is synthesis. We're taking things and putting them together, right? And X is the enzyme. So these are simple sugars. These are like the building blocks that build this huge carbohydrate named starch. Let's look at protein. Protein is going to look a little bit differently, but it can be shown in multiple ways. So let's look at digestion, right? So proteins are usually really long strands of amino acids, kind of like beads. And in our stomach, right, our stomach enzymes are going to break all these apart into individual amino acids. Cool? Those amino acids will go to our bloodstream. They'll be delivered to our cells. And our cells will then take these same dots. Of course, these are different shapes now, but it's kind of showing you that these building blocks are amino acids. And we will build our own protein from it. Of course, this is the job of an enzyme, right? To kind of glue those things together. We didn't identify where the ribosome was inside of the cell. So we're going to take a moment to show us how enzymes are actually made by our body. So our body makes enzymes in each of our cells. So we don't have like an organ that makes enzymes. All of our cells make enzymes. Where does that happen inside of the cell? That happens in the ribosome. So the ribosome is actually going to be number 1. Okay. It's these tiny, 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 tiny little dots. those are all ribosomes. So ribosomes are very, very tiny. Okay, so amino acids are taken to the ribosomes. They're going to be put together to build protein for the body. For example, right, enzymes are protein. So if you eat a steak, you're going to break that down into amino acids. Those amino acids are going to your cells. Your cells will put them in the ribosome and then make your enzymes. So what we need to remember, ribosomes are the site of protein synthesis, and they are an organelle inside of our cells. Let's talk about what happens when enzymes change shape and what factors really have a big impact on enzymes. So the two factors are temperature and pH. Okay, so enzymes are very sensitive to temperature and pH. They have a perfect temperature and pH where they work the best. And that is called optimum. And optimum is always at the very, very, very top of the graph. That is where the enzyme is perfect shaped and works the fastest. When we start to get too low, like down here, we see that enzyme actually changes shape and doesn't work anymore. And when it gets really hot, it does the same thing, right? If we go above to that fever about 105, we see that enzyme has completely changed shape and can no longer work because this is the original shape, right? So we're going to highlight the optimum temperature, and pH is the point at which enzyme has the best shape and works the best. So what happens, and let's just take a look and let's give that a word, right? If the temperature and pH are not at the optimum, and if they're too high or too low, the shape of the enzyme changes and won't be able to function. And this is called denatured. So I like this picture right here, kind of showing us that through heat and pH, we kind of see this denatured enzyme here where it's completely almost melted. What's important to note is that each enzyme has a different optimum pH and temperature. So let's look at a couple human enzymes here. So pepsin is an enzyme that digests protein in the stomach, and trypsin is an enzyme that digests protein in the intestine. So they're different enzymes, right? And notice that in the stomach, the pH is about, this optimum pH right here is about 3. And then the optimum pH of trypsin is just between like 8 and 9. Okay, so they have a different optimum pH despite them being in the body. And that's because the pH is different, or the acidity is different in the stomach than it is in the intestine. So what's interesting is if pepsin moves from the stomach to the intestine, it actually denatures completely because it's in two, it's not in its correct range, right? So if we were in the intestine, which has a pH of about between 8 and 9, but this is an optimum pH of 3, it's going to completely denature when it hits the intestine. And that's not a bad thing. That's actually supposed to happen. All right, let's talk about some common examples of how they talk about enzymes on the Regents exam. So they love to mention that Venus flytraps produce an enzyme in order to break down flies. They always talk about that. Another cool example is that spiders have enzymes in their venom that will break down other organisms. So they like to talk about that. They also like to talk about bacteria that live in super, super cold environments and really, really hot environments, and how they have different enzymes that will denature at a much higher temperature or a much lower pH. And then last but not least, they do sometimes talk about this enzyme called catalase that breaks down hydrogen peroxide. So just keep a heads up that that's a common enzyme that they do talk about on the exam. All right, let's walk through a couple practice questions pretty quickly. So it's talking about the pH of the internal environment of lysosomes is approximately 4.5. While the pH of the surrounding cytoplasm is approximately 7. The average pH of the human stomach during digestion is approximately 2.5, while the average pH of the small intestine during digestion is about 8. And it shows us a graph of the effective pH on enzyme activity, and these letters are different enzymes, A, B, C, and D. And it says, which enzyme functions in a pH most similar to that of a human stomach enzyme? Well, the first thing we need to do is find out what the pH of the stomach is. And of course, it tells you up here. So it says it's about 2.5. So we need to find the enzyme that we think is like the optimum at 2.5. So I would say that the optimum for this would have to be A, right? Because that's kind of between 1 and 7. The optimum is between 1 and 7. And that's really the only option. So A is the enzyme that would function most. I also wanted to show you a similar question, just because these can be kind of difficult. So this is a similar question. Okay, it shows you 4 enzymes. Okay, pepsin, papain, cryptomycin, and cholinesterase. Okay, and then it gives you the pH range of each part of the digestive system. So it shows you the mouth, the stomach range, small intestines, and large intestine. So let's see, which enzyme would most likely function in the mouth? So the mouth has a range of 6.5 to 7.5. So we need to figure out which enzyme has an optimum in 6.5 to 7.5. So let's see. Well, let's see. This one has about that, right? Let's see. So the mouth is 6.5 to 7.5. So we know it can't be pepsin, right? Because the optimal would need to be here. Papain, it would need to be there. So let's see. I would say it would definitely be chymotrypsin. Spelled that wrong a little bit there, but I guess the point across. Cool. So I just wanted to show you a couple of examples for those more difficult questions. All right, we've talked about enzymes, and I will tell you in the iPuzzle what you need to do next.